Hello students, today we will be discussing about dihybrid cross. So a dihybrid cross talks about two pairs of contrasting characters. Here we have taken the seed shape and seed color for pea plant, Pisum sativum, as studied by Gregor Mendel, right? Talking about the seed shape, the dominant character being capital R, and the recessive is wrinkled, small r we have taken. The seed color, yellow being dominant, we have considered capital Y to be the dominant one, and the green, the recessive one, to be small y. So we can find out that if we have taken the parental generation as capital R, capital Y being a dihybrid cross, so the small r and small y is the recessive. We get the gametes out of them, and in the F1 generation, we can see in the Punnett square, we get 100% round and yellow seeds. So the phenotype for the F1 generation stands to be 100% round and yellow seeds, all round and yellow seeds, correct? And then the genotype talks about capital R and capital Y small y. So that is heterozygous round and yellow seeds for a dihybrid cross in the F1 generation. Now the next step that we follow is to find out the F2 gametes. And we proceed accordingly. We go for selfing the F1 heterozygous that we have got. We self it and we check that capital R crossed with capital R, capital Y small y, crossed with capital R and capital Y small y, we get the F2 gametes. How to find out the F2 gametes? We have to place the similar alleles to one side of the Punnett square, and then we get the gametes for the F2 generation to be capital R, capital Y, small r, capital Y, capital R small y, and small r small y. So these are our F2 gametes. Now we will place them in a Punnett square. Now the Punnett square that we look for in case of dihybrid cross is a 16-box square. 16-box square. Let's see how it comes up. So here is the F2 generation, the Punnett square, 16 boxes Punnett square that we take up for the F2 generation in case of a dihybrid cross. As you can see, we have framed the gametes and now we have placed the gametes accordingly. We have placed the first series of gametes on the top, on the upper side, and the similar series of gametes on the other side, male and female gametes. Now, how do we form this Punnett square full? Here we can see that we have taken capital R and capital R, placed them side by side, and then capital Y and capital Y, place them side by side. So round and yellow it represents. And then we have kept capital R and small r with capital Y and capital Y here. Taking capital R and capital Y and small y, we always place the recessive allele towards the righter side and the dominant allele in the front. That is the format that we always follow. It helps us to understand the dominant character as well as the recessive character. As per the law of dominance, we know that the dominant character always expresses itself, so the dominant character is placed in the front. Okay. And then finally the small r here and small y crossed with capital R, capital Y comes out to be capital R small r and capital Y small y. Correct? So in this way we fill up the whole box of the F2 Punnett square, and we can see there are 9 round and yellow seeds. If we go to the boxes, we can see there are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 round and yellow seeds, followed by 3 wrinkled and yellow, 1 wrinkled yellow, another wrinkled yellow, and another wrinkled yellow here. Okay, followed by the round and green, 1 round green over here, another round green over here, another round green over here, 3 round green, and finally 1 the wrinkled and green at the end of the boxes. So 16 boxes square we got. Now we need to find out the phenotype for the same. So how the phenotype goes? The phenotype, that is the expressed character, the externally observable characteristics for the F2 generation here comes up to be round and yellow seeds 9, as we checked there, wrinkled and yellow seeds 3, as we found from the square, round and green seeds 3 again, and wrinkled and yellow seeds to be 1. So the phenotypic ratio comes out to be 9:3:3:1. So whenever we are asked a question in the boards regarding the dihybrid ratio, in case of dihybrid cross, the phenotypic ratio, we mention it to be 9:3:3:1. Okay. Now next we pass on to the genotype, the genotype of the F2 generation. So while finding out the genotype, we need to place the box and find out each and every box, how many times each and every genotype has occurred in the Punnett square box. From that we have framed out capital R, capital Y has been seen only once, capital R, small r, capital Y twice. And then like in the same series, we have framed out for each and every box and found out these numbers. And hence the genotypic ratio of F2 generation appears to be 1:2:1:4:2:1:2:1:1. So this is the complete dihybrid cross in case we have taken 2 pairs of contrasting characters as proposed by Mendel. And here, as already said, that we have taken the pea plant, Pisum sativum, 2 pairs of contrasting characters for Pisum sativum, seed color and seed shape. So this completes the dihybrid cross when we have taken 2 pairs of contrasting characters. Hope this is helpful. Thank you.